Hi, my name is Wes. I'm a postdoc in Manus Donahue's lab at Vanderbilt University Medical Center. I'll be talking about detectability of the white matter perfusion across flow territories, looking at different hematocrits and bolus arrival times. So previous studies have shown white matter perfusion to be difficult with reasonable scan durations. This is primarily due to low cerebral blood flow and longer bolus arrival times, limiting the signal in an already limited signal to noise ratio imaging modality. And we sought to revisit this issue across a range of inversion times as well as hematocrits, and to test the different hematocrits or the effect of different hematocrits on white matter perfusion detectability. We recruited sickle cell disease patients, which has already been covered multiple times. I won't go into too much detail, but it's an inherited genetic disorder that affects the production of hemoglobin. That disproduction of hemoglobin or misproduction of hemoglobin leads to hemolysis, downstream anemia, and furthermore, decreased arterial oxygen content. And as a result, as has already been covered during this meeting, cerebral blood flow is upregulated to maintain oxygen delivery. And you can see that here, where the cerebral blood flow of the sickle cell anemia patient is much higher than that of the control participant. And so, although this adaptation is effective, the sickle cell disease patients have an increased risk for silent and overt infarcts, as you can see in the bottom right panel. This is a combined study of over 400 subjects, where you can see the prevalence of silent cerebral infarcts increases linearly with age for the sickle cell disease patients. And furthermore, there seems to be no plateau in this plot either. Additionally, it's been estimated that around 90% of silent cerebral infarcts occur in the border zone regions of the white matter, which is particularly difficult when considering arterial spin labeling, because those are the regions in which it is least reliable due to low cerebral blood flow and long bolus arrival times. And this image in the bottom left is the study that found the estimated 90% of silent cerebral infarcts in the border zones, and this is just a heat map of their patient population, silent cerebral infarcts. And so we hypothesized that due to the increased cerebral blood flow, shortened bolus arrival times in sickle cell disease relative to controls, we predicted that arterial spin labeling will have an increase in detectability of white matter cerebral blood flow. Furthermore, as sub-hypotheses, we predicted that cerebral blood flow will be negatively correlated with hematocrit, so increased as hematocrit lowers. And additionally, that bolus arrival times will be shortened for lower hematocrits as well, so inversely correlated. And the impact of this work, if white matter perfusion quantification is determined to be feasible in sickle cell disease patients, it could be investigated further as a biomarker for infarct risk. And so just kind of like a little quick note about the sickle cell disease patients that we recruited. We recruited adults with sickle cell disease, all variants, but we have excluded patients with overt strokes as well as focal neurological deficits. In addition, moya-moya vasculopathy, since the decreased cerebral blood flow with vasculopathy would make it even harder to detect white matter perfusion. And then in addition, we collected healthy age-matched controls. And so to test the detectability of white matter perfusion, we used a multi-inversion time pulsed ASL sequence where we got 25 repetitions of 16 inversion times interleaved between two sequences, and equally spaced inversion times between 200 and 3200 milliseconds. And so this basically consisted of our first sequence where we got 8 inversion times equally spaced between 200 and 3,000 milliseconds, so spacing of 400 milliseconds. And then the second sequence, we just interleave that in between those. So from 400 to 3,200, still with the 400 millisecond spacing. The resolution was 3 by 3 by 7 with 9 slices, and then we use a look-locker readout to achieve the number of inversion times. And furthermore, especially as we're reviewing the bolus arrival time results, the labeling volume was only 100 millimeters proximal to the imaging volume. And furthermore, the acquisition time, including the M0 collection, was about a little under 7 minutes. And additionally, we collected an anatomical T1 for segmentation and registration. And so to quantify cerebral blood flow and bolus arrival time, we have these nice, relatively high temporal resolution time series for perfusion. And so we plugged it into FSL basal. As you can see on this top right, we have fit to a example voxel where this is the Buxton 3-stage model. We have blood flow in, T1 decay in the tissue, and then the measured blue in the background. Furthermore, we corrected the arterial blood T1 for the hematocrit collected on the same day. In addition, we used a look-locker tissue T1 correction, as already has been shown. And then furthermore, since we're trying to test this across a number of different flow territories, we used a arterial flow territory atlas that was recently published right here by Lou and colleagues in Science Data, where we looked at the cerebral blood flow and bolus arrival times in the anterior middle cerebral arteries, as well as the border zone territories. Due to the limited field of view of our PASL sequence, we were unable to characterize the posterior cerebral artery flow territories. And then lastly, we tested the correlation between whole gray matter and white matter, cerebral blood flow, and bolus arrival times relationship to hematocrit. And to calculate the perfusion detectability, we used the same method as Van Osch and colleagues in 2009, where we looked at the T statistic of the perfusion measured. So, for example, this is one voxel where we measure perfusion 25 times over the 25 repetitions, right? And so from that, we get a distribution of the perfusion measured. And if we compare that with 0 perfusion detected, we get some type of estimate of our perfusion detection. And additionally, we can convert this T statistic into a p-value and consider where perfusion is significantly detected. And we did this on the group level in each flow territory measured, as well as over the whole tissues. And we considered the mean minus 1 standard deviation of the group above p less than 0.05 to be significant across the group. So this is the cohort that we collected. We collected 35 sickle cell disease patients and 50 healthy age-matched controls. There's no difference in sex or age. We did not match for race. And then the sickle cell disease patients are obviously neither. And so this is just a representative perfusion-weighted time course volume or slice in each group. So starting on the top left, this is our earliest inversion time, the 200 milliseconds, and then keep following left to right, and then down here all the way to 3,200 milliseconds. So you can see the blood flow in to the arteries, into the tissue, and then furthermore decay with tissue T1. Additionally, even on the qualitative assessment of a single slice, you can see the blood arrives earlier in the sickle cell disease patient, and furthermore, the intensity is higher. So it's relatively, it's nice to be able to appreciate these things qualitatively on just the images that we get off the scanner. before any type of absolute quantification. And so moving on to the absolute whole group quantification of cerebral blood flow and bolus arrival times, we see that this is all subjects transformed into M&I space and then averaged, just to appreciate the differences between the two. And so, as we can see, the mean cerebral blood flow is higher in the sickle cell disease patients. Furthermore, as anticipated and known, the white matter is lower relative to the gray matter. But furthermore, the bolus arrival times, you can see the healthy controls are much longer, as denoted by the red. When compared with the sickle cell disease patients, much shorter. And furthermore, as anticipated as well, the gray matter is somewhat shorter than the white matter. However, I think the difference between these was less than we were anticipating as well. And so moving on to the quantitative results across the group, we found what other studies have found in that the cerebral blood flow was significantly elevated across all ROIs measured in tissue types. But the bolus arrival time was only significantly shortened in the white matter, which could be due to power and maybe just not enough of a effect size in the gray matter. And then when we plotted the T statistics I was describing earlier in each group, what we found, so first I'm going to focus on this magenta line. So when we look here and here, we see that the healthy controls that we recorded were significantly detected as a group, so mean minus standard deviation at 1400 milliseconds. However, the sickle cell disease patient group was significantly detected between 800 and 1800 milliseconds. A much wider range. And then probably most critically, we found that the sickle cell disease group, when we look at the border zone region of the white matter, so this red line here, we can see that the 1400 millisecond time point for the sickle cell disease patients was significantly detected across the group. So yeah, especially when we're talking about the infarct risk assessment, it was good to see for sure. And then furthermore, when we looked at our kind of like side hypothesis with the cerebral blood flow and bolus arrival times relationship with hematocrit, we found the correct relationship, but not significant relationship between cerebral blood flow and hematocrit in the sickle cell disease patients. But we found a very strong relationship between the gray matter bolus arrival time and the hematocrit, and that was furthermore matched in the white matter, if not stronger in the white matter in terms of bolus arrival times. And so in summary, we found and reproduced what previous results have shown, that cerebral blood flow is greater, bolus arrival times are relatively shorter in sickle cell disease patients, and the white matter perfusion was better detected in sickle cell disease patients. Furthermore, between 800 and 1800 milliseconds, the whole tissue average was significantly detected. And even in the age-matched controls, we found at 1400 milliseconds, significant detection of perfusion. And what I think is most critical, we found border zone perfusion significantly detected in sickle cell disease patients at 1400 milliseconds. And then finally, we found bolus arrival times were better related with hematocrit than cerebral blood flow. This could be indicative of our more heightened sensitivity with the pulse. So we're labeling very short, and the sensitivity to those arrival times is much greater, we think. But in addition, this could be evidence of blood flow velocities upregulated in response to hematocrit declines. And so in conclusion, white matter perfusion detection is much more feasible in sickle cell disease patients than controls. This is especially true in the border zone regions of the white matter, which may prove critical for assessing infarct risk, especially longitudinally. And then bolus arrival times were found to be more sensitive to hematocrit in sickle cell disease patients as opposed to cerebral blood flow. And so we were talking about kind of like the shunting hypotheses that was discussed yesterday. I think bolus arrival times, arterial arrival times, maybe some type of difference between those might prove to be an interesting metric in terms of testing that hypothesis relative to oxygen metabolism and extraction in this patient cohort. So yeah, I would like to thank my advisors, Amanda Sonahue and Lori Jordan, as well as my team, funding for this work, and our volunteering participants.